Welcome, Fact Fanatics, to another exciting episode of G Fun Facts Online. Today, we're diving deep into the squishy supercomputer between your ears, your brain, and uncovering the secrets of how it stores a lifetime of memories. Ever wondered how a fleeting experience transforms into a cherished memory you can recall years later? The answer, believe it or not, lies in tiny molecular machines working tirelessly within your neurons. Get ready to have your mind blown. Our journey into memory starts with something called synaptic plasticity. Sounds complex, right? But think of it like this. Your brain is a vast network of connections, like an incredibly intricate roadmap. Synaptic plasticity is the brain's ability to redraw that map, strengthening some routes and weakening others, based on your experiences. This constant reshaping is how we learn and remember. And who are the crucial traffic controllers in this process? Tiny protein pores called ion channels. Now let's zoom in on one particularly fascinating type, calcium ion channels. Specifically, a channel known as Cav 2.1, which is abundant in our brains, has a kind of molecular memory all its own. Imagine this. When a neuron gets zapped with electrical signals repeatedly, these calcium channels can actually change their shape. They can enter what scientists call a declutched memory state, making them less likely to open. This change, this memory, can last for several seconds in a single channel. That might not sound like much, but when you have countless channels across many neurons all doing this, the effect adds up. This accumulation can reduce communication between specific brain cells, leading to changes in the receiving neuron that can last for hours, even days. It's like a tiny whisper that, when repeated enough, becomes a lasting instruction. These short-term molecular memories are stepping stones, contributing to much bigger, long-lasting changes in the brain, like pruning away weakened connections, ultimately helping to cement those lifelong memories. And get this, these channels are incredibly versatile. Researchers have found they can adopt almost 200 different shapes depending on the electrical signals they receive. Talk about adaptable. But calcium channels aren't working alone. Let's introduce two other crucial players in the memory game: NMDA receptors and AMPA receptors. Both of these are activated by a chemical messenger called glutamate. NMDA receptors are super cool. They're like coincidence detectors. They need two things to happen before they open and let calcium ions flood in. Glutamate has to bind to them, and the neuron needs to be sufficiently electrically excited. This calcium influx is a critical trigger for processes that strengthen or weaken synapses, known as long-term potentiation, LTP for strengthening, and long-term depression, limited, for weakening. Both are vital for carving out memories. Then you have AMPA receptors. These guys are also essential for the actual expression of these synaptic strength changes. The number of AMPA receptors at a connection point can change dynamically, making the connection stronger or weaker. So it's this intricate dance between NMDA and AMPA receptors that initiates and maintains the changes in our brain circuits that underpin memory, and the list goes on. Potassium channels, the most diverse group of ion channels, also play a huge role. Some of them are activated by the calcium we've been talking about, and they help shape how neurons respond and how strong the signals between them are. Recently, scientists have discovered that these different ion channels don't always act solo. They can team up to form complexes called channelosomes. Think of them as supergroups, where NMDA receptors and other channels like BK potassium channels work together. These complexes allow for incredibly precise control over neuron function and how our synaptic connections change, coordinating their activities for maximum effect. Now, why is all this tiny molecular detail so important? Well, when these ion channels or their interactions go awry, it can lead to various neurological and neurodevelopmental disorders that affect learning and memory. So the more we understand about these intricate molecular mechanisms, the closer we get to understanding the very essence of cognition. This deeper knowledge isn't just fascinating. It opens up exciting possibilities for developing new treatments for memory-related disorders by targeting specific aspects of how these ion channels work. So the next time you recall a fond memory, take a moment to appreciate the incredible, microscopic ballet happening inside your brain. Millions of tiny ion channels, opening, closing, changing shape, and communicating, all to transform your experiences into the memories that make you who you are. It's a universe of complexity within us. That's all the time we have for today on G Fun Facts Online. I hope you're leaving with a newfound appreciation for the microscopic marvels that make your memories possible. Join us next time for more amazing facts. Stay curious, everyone.